people that put this thing together, because to me it's one of the best things that JSU does. It gives students the opportunity to actually present some of the work, especially those students who are getting ready to go to grad school, and more specifically at other schools, because I personally feel like this gives them a leg up, an opportunity that many other students going into grad school don't have. It does me great pleasure to actually introduce Kaila Brown. Kaila is what I classify as, this is what we classify as sort of a transdisciplinary research project, because we're really big on crossing those disciplinary boundaries. Kaila is a minor in biology, major in geography and spatial analysis, which I think is a perfect marriage. I haven't convinced all of the biology people of that yet, but I think it is anyway. And so what she's going to be looking at here, and I'll go ahead and give away the title since it's up there, Spatiotemporal Investigation of E-coli Contamination of Tributary Creeks from Tallahatchie to the Coosa. And this is a culmination of research that's been done by a number of people, including the Water Watch folks in this particular area, taking samples of local water, the streams in a localized area, and ultimately how these are going to affect the downstream situation, such as flowing into the Coosa River, ultimately, because we are all part of that Coosa watershed. With that, Kaila?
As Dr. Morgan said, I'm currently working on a project that we've called Spatiotemporal Investigation of E. coli Contamination of Tributary Creeks from Tallahatchie to the Coosa. I'd like to start off by saying thank you to the Jacksonville River monitors for collecting the data that was used in this project, as well as Dr. Case for helping run statistics on the data, and Dr. Morgan for supporting this research. The National Water Quality Inventory ranks pathogenic bacteria as one of the most widespread pollution problems in our nation's rivers and streams. In an attempt to determine pathogenic pollution problems and potential origins of bacterial contamination in local tributaries of the Tallahatchie Creek in Jacksonville, Alabama, we conducted a one-year preliminary study. E. coli is a species of fecal coliform bacteria that is specific to the fecal material of humans and other warm-blooded animals. The EPA recommends E. coli as the best indicator of health risk for water contact in recreational areas. An indicator species serves as a measure of environmental conditions that exist in a given locale. Although they are generally not harmful themselves, their presence in streams suggests that pathogenic microorganisms might be present and that swimming and using the water in the area can be harmful to health. Sources of contamination to surface water include on-site septic systems, water treatment plants, domestic and wild animal manures, and storm runoff. In addition to the possible health risks associated with the presence of elevated levels of fecal bacteria, they can also cause cloudy water, odors, and increased demand in oxygen. Water contamination can lead to recreational water illnesses. These are caused by germs spread by swallowing, breathing in, or having contact with contaminated water in water play areas, lakes, rivers, and ocean. About 91 million people over the age of 16 swim in our nation's oceans, lakes, and rivers, and a total of 81 water illnesses outbreaks have been reported to the CDC since 2010, and this has affected close to 2,000 people. So sampling was conducted in 3 tributaries of the Tallahatchie Creek from March 2014 until February 2015. Our sample site X is located in Germania Springs Park, just north of the Jacksonville State University campus. The site is used as a public park area that hosts ball fields, a small dog park, a playground and pavilion area with on-site restrooms. Site X water begins at Germania Springs Head and dumps into Flat Tire Creek, and then later enters the Tallahatchie Creek. Site Y is located near Jacksonville Recreation Center and is also classified as a public park area. The Ladiga Trail runs alongside and across the creek sampling site known as Haynes Branch. Site Z is a continuation of Haynes Branch, passing under Highway 204 east of Gadsden Road near a local barbecue restaurant Both sites Y and Z flow from Haynes Branch directly into the Tallahatchie. Also, the testing sites flow near the Jacksonville Water Treatment Facility, but the water is not treated by the plant. So, under Alabama Water Watch Protocol, bacteriological tests were conducted on a monthly basis. Certified water monitors perform bacteriological sampling at each of the sites on the first Saturday of every month. Monitors collected air and water temperatures from the site, as well as 1 milliliter water samples from 3 different locations within each of the testing sites. The samples were then transferred into bottles of media and taken back to the lab to be plated. Petri dishes were labeled with testing site information, and then the media bottles were poured into the corresponding Petri dishes. They were set to gel, and after the media gelled, it was placed in an incubator at 29 to 37 degrees Celsius for 30 to 48 hours. After incubation, the plates were removed and the E. coli colonies were counted and recorded. Bacterial concentrations were calculated using the formula E over V times 100 equals C, where E is our number of E. coli per plate, V is our volume sample, and then C is our total number of E. coli per 100 milliliters of water. The EPA establishes that E. coli should not exceed 200 colonies per 100 milliliters of water. The site totals were averaged for the entire year, and E. coli colonies per site were, as you can see on the graph, Site X averaged a total of 19 colonies per 100 milliliters over a 12-month period. Site Y averaged slightly below our area of concern at 150 colonies per 100 milliliters, and Site Z showed the most concern above average at 250 milliliters over a 12-month period. The site totals were averaged on a seasonal basis as well, with Site X showing colonies in spring and summer at well below the area of concern. Site Y is also below our area for all the seasons, with summer and fall being the highest levels. These levels were not far from our area of concern, so they could be a potential threat. And then Site Z showed the most concern with levels of E. coli at or above our area of concern in spring, summer, and the fall seasons. The site monthly averages were calculated as well, with Site X showing mostly levels of 0 with a small spike in April and then a larger spike in June. Site Y has colonies showing up every month but reaching concern in August, October, and December. And then we had Site Z showing colonies every month except for December, and a relatively constant above average total for the months of May through October. This shows a seasonal temporal elevation for our Site Z. Statistical analysis support elevated levels of seasonal counts in the summer and fall at the 0.05 level. Site X is statistically significant in summer, although it is well below our 200 level. It shows higher spikes. They're not at the 0 level, and so this shows some concern for the summer and the fall seasons. And Site Y and Z showed statistically significant values in the summer as well as the fall. Pathogenic pollution is cumulative. It is expected that the waterways continue to become more polluted as they flow towards the Coosa River. The next step in this research is to find our potential sources of contamination. Ground truthing and high-resolution Landsat ETM imagery, along with the evaluation of the agricultural calendar and the agricultural calendar, as well as the weather history, will be used to evaluate land use and land cover in our drainage area of the testing sites. A review of seasonal factors such as temperature as well as potential sources of pollution, is being conducted to better understand the risk of human exposure to E. coli in the recreational freshwater areas. Here is a larger view of the map that shows our testing areas. You can see how they connect and eventually flow into the Tallahatchie. And then I couldn't fit the whole map on the screen, so later they flow into the Coosa. So this, as you can tell, affects a large portion of Calhoun and other counties in central Alabama. Are there any questions? Yes, ma'am.